number of people you can have wire things. If you have a technology like photolithography that every year can get smaller by a factor of 2, then the number of transistors that you get grows exponentially by factors of 2 every 18 months or so. And so what is missing, or has been missing from biology, is that we're really building biology, or we have been building biology, the way that we used to build electronics, by wiring things up by hand. So what's the future for biology? The future for biology is something that we call a biofab, and this is something that we've been working on and other people have been working on. And it's really to apply some of the same technologies that we use from semiconductor, but instead of patterning silicon, now we're going to pattern biology. And so we have a technology that is based on lithography, but it's a little bit different because in this case, instead of having the photons land on a surface and develop a photoresist that we would do for semiconductors, in this case, where the photon lands, it changes a chemical property of a nucleotide base to either have a protection group or not have a protection group. And so wherever a photon hits, it deprotects that local nucleotide and allows us to add the next nucleotide. And by doing this, we can really go from building things that are 100 or a few hundred nucleotides in length to building millions of little pieces of DNA, enough DNA on a single chip. By the way, these chips cost now about $400. We can build enough DNA on this chip to code to build all the DNA for an entire organism. Okay. So the next step is that we need to start to put these pieces of DNA together and to create things that are long enough. So now we have enough little pieces that are comprise all the different pieces we need for an entire genome, but now we need to put them together into a single long piece that can become a full genome. And this is a whole nother area. I'm not going to go into all the details of it. There are many issues that are involved because all of the small pieces of DNA have errors in them. And we need a way to create a single long piece that has no errors. And so in our laboratory and working with other people, we've developed such a technique, what we call error-correcting synthesis, that allows us in little devices like this, these are little microfluidic chips, that allow us to put pieces of DNA that come from the DNA chip into these and assemble them automatically into long, perfect pieces of DNA. What are the things that we can what are the things that we can start to build with this, and why is biology really so exciting and so profound? So I just want to very quickly give a little taste for this. Many people don't realize that biology really can be programmed in the way that we can program a computer chip. The very first thing when you're building a new computer chip to test out the factory that builds the computer chip is to build a simple circuit called a ring oscillator. A ring oscillator is a couple of transistors where the output of one goes to the input of the next and so forth, and the very last one is fed back to the first one. And so what happens in such a circuit is you get a sine wave. That circuit oscillates. It's called a ring oscillator. Biology, we can build exactly the same kinds of circuits. In fact, we say that the logic within biology is universal, meaning we can create anything that we can create out of silicon, we can create from biological parts. This is in fact such a ring oscillator. It looks exactly like 3 transistors. This transistor, this gene, creates a protein which turns this one off. This one creates a gene which turns this one off, and this gene creates a protein which turns this one off. And so you just start this in an abundant state of this TET protein. You have a lot of TET protein that turns this one off, which allows this one to be on. If he's on, he creates enough protein to turn him off. Now he's off, that allows him to be on, and so forth. And if you tie this to another protein which glows green when one of these is on, let's say this one, and you look at this with a camera, you'll see exactly the same kind of behavior that you have from the electronic circuit, meaning a sine wave. The only difference is that in the normal electronic circuit, the sine wave is constant like this. But here, inside this bacteria, the sine wave is growing. The sine wave is getting bigger and bigger. Why is it getting bigger and bigger? Because biology does something that circuits don't do. They replicate. And the amazing thing is that even though I only created this circuit once in this first bacteria, when this bacteria divides and creates a second one, it also creates a copy of that circuit. And it creates that circuit in the same phase. And so now the sine wave gets a little bigger. And then those two divide, and they create the next multiple of those circuits. And so I only have to create that circuit once, and that's the profoundness of biology that I have known no other discipline of engineering, this ability to self-replicate and to grow. We can create very complex circuits. I'm not going to go into all the details of it. We can do other profound things. We can cure diseases. Malaria is a disease that kills, unfortunately, many people, a few million people a year. There's a cure for malaria. That cure comes from a tree called a Chinese wormwood tree, and it creates a very small molecule called artemisinin. We can clone that pathway out from artemisinin, from the wormwood tree, and put it into a bacteria, and try to create this small molecule which cures malaria. The problem is that even though I told you the genetic code is the same in all the organisms, the dialect is a little bit different. And so when I clone out that pathway and put it into a bacteria, it's not quite correct. The concentrations of the different proteins aren't correct. And so what I'm forced to do is sequence that and then translate it into the dialect that the bacteria speaks. Unfortunately, we're not very good translators. We don't know how to do that exactly. And so what we have to do is build lots of different variants of that. Right now, to do a project like this takes about 5 years and about 50 postdocs to do a project like this. When we start to bring this biofab online, we'll be able to do projects like this, the first synthetic biology engineering projects. We'll be able to do them in a few months. I'm going to, you have to give me a timing so I don't, this is maybe the most aggressive project in synthetic biology. This is a very large program in the United States that's funded by the National Science Foundation called SynBirth. And the idea is that living inside of all of us are little bacteria. They live in our mouth. They give us cavities. That's why we have to brush our teeth. And they live inside of our stomach. But we don't have any interaction with them. We didn't put them there. We didn't do anything to make them more useful to us. So the idea is if we could reprogram these bacteria so that instead of just being a bacteria that has no particular benefit to us, what if we could put a little sensor onto this bacteria, a little receptor that is a receptor for a cancer marker, a cancer protein, for instance. And so that these bacteria would live inside of us, and when it sees a cancer marker, it might signal us in some way. It may make a fluorescent protein, for instance, which will come out in our urine, and it's an indicator, a very early warning system that, we have something that we should look at inside of us. So how do we start to do this? Well, there's a extremely interesting collection of different parts. Where do you get such a sensor for a cancer marker? Well, if you go to this place, parts.mit.edu, you can find thousands, about 30,000 different parts that you can add to your own bacteria. Sensors, various different pieces of logic. For instance, you might want a piece of logic that says only when sensor A and sensor B both detect a marker, only then create a dye. That would be a logical and. So I can go to my parts list and start to take out the sequence for that part and then program that into the bacteria. This is the other big area in synthetic biology, is biofuels. you may know that biofuels really have a lot to credit the first president of Israel, Chaim Weizmann. The organism that people are very excited about is an organism called Acetobutylicum. This is an organism that Chaim Weizmann discovered in the early part of the 1900s, and about $1 billion of investment is going into engineering this organism to create a next gen of biofuel called butanol. And again, to do that, we need to change about 1% of the genome in order to make organisms that can produce this at a high efficiency. So I want to come now to the second area, the next step up of changes, which is changing the genetic code. So this is a project that exists. It's a collaboration between my lab at MIT and a colleague of mine at Harvard Medical School named George Church. And this is a NSF project that we have, which is called E. coli. So bacteria, a certain class are called E. coli. This is E. coli. And what we mean by that is rewriting the genetic code. What do I mean by rewriting the genetic code? Well, every 3 nucleotide bases that I have in a genome codes for a particular amino acid. So for instance, if I have a code UUG, that codes for leucine. You can see that there are a number of things that code for leucine here. And I don't have time today to tell you why it is that there are several different things that code for leucine. But what I mean by changing the genetic code is, what if I make UUG, this UUG, code not for leucine, but for some other amino acid? All of biology that we've discovered across the world not only has the same genetic code, but it also builds proteins with the same 20 or 21 amino acids. On my shelf at MIT, I have a catalog of about 2,000 non-natural amino acids, synthetically made amino acids. I might like to have an organism that can build with completely new materials. I might like to be able to make drugs that have a protein that is built out of different kinds of amino acids, maybe fluorinated amino acids, that have completely different properties than what current biology knows how to build with. To do that, I'd like to create an organism that has a different genetic code. So this is a very difficult project that we engaged in, and we're I can just give you a status update. We're close to doing this. This is the organism that we're working on right here. It's called MG1655. It's about a 4.5 megabase organism. And the very first thing that we're doing is we're freeing up one of these stop codons. The genome has a certain sequence that tells the ribosome where to stop. So the ribosome moves across a piece of mRNA and creates a protein, and it needs to know where to stop, and to do that it's got a certain stop code. Happens to be 3 of those stop codes, and what we're doing is we're freeing up one of those stop codes. This TAG that you see here, if you have extremely good eyes, is one of the stop codes. And what we're doing is everywhere in this genome that we find a TAG, we're reprogramming it to read differently TAA, which is also a stop. That'll produce for us a genome that has 1 free codon that we can use for a different purpose. And there's about 326 of those changes, and we've made all of those changes now. And we're at the point where we're starting to combine bacteria. Each bacteria that we've made have a different change in them. And so now what we need to do is take a bacteria A and a bacteria B, each that has 1 different change, and combine them together. And this is Nogea to what Rabbi Rappaport is going to talk about. Bacteria know how to combine genetic material. They have a process called conjugation, where they can create a small protein tube like this and exchange genetic material from one bacteria to another. Very similar to animals to macroscopic mating, but this is done on a single cellular basis. And so you can see right away where some of the first issues start to arise is if we create a certain bacteria A, a certain bacteria B, are there any issues about combining those? Does Alaka have anything to say about that? Or no, maybe everything is mutar. That's not the answer. Okay. So I want to make sure we save time for the most important part of this day, which is the halachic discussion. So I just want to tell you about the last areas of effort, which are really to create a whole genome from scratch. So that is also a project that is well underway. The smallest organism that we know of has about 500,000 bases in it, but a more typical organism is similar to the one that I showed you. It has about 4.5 megabases, really to make an organism that can live generally in general environments. You need something about 4 million bases. Inside of us are 3 billion bases. Inside of a corn plant is 6 billion bases. You can think about, for a homework problem, why corn has twice as many nucleotide bases coding for corn than code for us. I can give you the answer if you want after the talk. But to make a bacteria, we need about 4 million bases. Right now, each base costs about 50 cents to make, and so that's about a $2 million project. When we start to bring this biofab technology online, we'll be able to bring that price tag down from $2 million. $2 million means only 1 or 2 labs in the world can work on that. When the biofab starts to come online, we'll bring that price tag down to about 1 cent per base. And so to build a bacteria will cost on the order of $40,000 or so. And the reason that this is an important halachic issue is that my prediction is that we will start to see an explosion of the number of labs and the number of individuals that are involved in these activities. Just as a status update, this is really the latest result. This is work from Craig Venter. By the way, they sponsored before they did this, they sponsored a study amongst different religions, Christianity, Judaism, and Islam. And they concluded that everything was mutar to do because religion didn't know about bacteria, it didn't know about genetic manipulations, and so religion is silent on this topic, and so everything is permissible to do. Of course, they didn't have somebody who had studied Masechet Kilayim, and so that's what we're here to do today. So anyways, I'm going to end here, and thank you very much.
כפי שסיים פרופסור יעקובסון, ניצבים אתגרים רבים לאור המציאות המדעית הזאת שהוא הציג בפנינו, כשהמציאות הזאת יוצרת שורה של אתגרים, לפחות עד כמה שאני הבנתי, שהם אתגרים חוקיים, פילוסופיים, אתיים והלכתיים. וכמובן לא נוכל לעסוק בכולם, אבל אני רוצה להזמין את ראש בית המדרש במכון הגבוה לתורה, הרב שבתאי רפפורט, לשאת דברים על ההיבטים ההלכתיים והפילוסופיים של המציאות הקסומה הזאת שהציג בפנינו פרופסור יעקובסון.
אז ברשות הנשיא, ברשות מורי חותני, שבעצם לא, מבחינתי הוא הדמות המודל לחיקוי לתורה ומדע, הוא בעצם מי שאחראי לשינוי בפסיקה בענייני רפואה והלכה בתורה ומדע, זה מורי חותני. באמת עשה שינוי גדול בדור שלם של פסיקה, חלק מזה, פסקים שמשה פיינשטיין כתב, ובאמת חידשו דרכים למחשבה. ובעצם, כמו בדור חדש, כן, צריכים להתקדם. והעיקרון הוא שלא ייתכן שהמדע יתקדם, תפיסת העולם תתקדם, ולא התורה. התורה לא תישאר במקום, זה דבר שהוא לא עולה על הדעת. והשם ניצוצות היה מכמה היבטים, ואחד מהם היה מדרש תהילים המפורסם על מוצאי שבת, שכאשר האדם חטא ביום שישי, ובשבת הוא היה בגן עדן, במין גן עדן, חשך עליו העולם במוצאי שבת, והוא אמר שהעולם אבוד ונכנסה עליו מיטה ואין לו שום תקווה יותר. אז הנוסח הרגיל של המדרש שמופיע במקומות רבים, שהקדוש ברוך הוא נתן לו שכל ולקח שני רעפים והקישם זה בזה ויצא מהם ניצוץ. אבל במדרש תהילים כתוב שאת השם של שני הרעפים זה אופל וצלמוות, והקישם זה בזה ויצא מהם ניצוץ. אז יש דברים שהם לא מובנים אולי כשלעצמם, מציגים אתגרים כשלעצמם, והניצוץ יוצא כשמקישים אותם זה בזה. השאלה הראשונה שעולה לדיון באמת בכל עניין שהוא מסוכן, שיש לו פוטנציאל גדול של סיכון, זה איפה אנחנו עומדים, ובאמת ההבטחה שלנו, התקן לאבטח את הטכנולוגיות, הוא השאלה של מה מותר לעשות ומה אסור לעשות. ובתורה בפרשת קדושים כתוב את חוקותיי תשמורו, בהמתך לא תרביע כלאיים, שדך לא תזרע כלאיים, ובגד כלאיים שעטנז לא יעלה עליך. הפסוק, המילים, 3 המילים, את חוקותיי תשמורו, מפרש הספרי חוקות שחוקקתם בשמיים ובארץ. זאת אומרת בעצם, שמור את העולם. אומר הרמב"ן, שהאדם צריך לשמור את העולם ולא לחדש בו מינים חדשים. והטעם בכלאיים הוא כותב, כי השם ברא המינים בעולם, ובאמת יש כאן הגדרה מדהימה של הרמב"ן לגבי חיים. השאלה מה נחשב חי ומה נחשב לא חי, זאת שאלה שהיא עולה כל הזמן לדיון, וכפי שפרופסור יעקובסון מסביר, הדבר החי, היחידה החיה הקטנה ביותר, הבסיסית ביותר, היא יחידה שיכולה ליצור את עצמה, להכפיל את עצמה. באמת זה מה שכותב הרמב"ן בהגדרה של חיים. כי השם ברא המינים בעולם בכל בעלי הנפשות, בצמחים ובבעלי נפש התנועה, ונתן בהם כוח התולדה שהתקיימו המינים בהם לעד. זאת אומרת, מהי הגדרת דבר חי? מה שיכול להכפיל את עצמו שיתקיים הלאה. וציווה הקדוש ברוך הוא בכוחם שיוציאו למיניהם ולא ישתנו לעד לעולם. אז יש פרוטוקול של תיקון שגיאות שטבוע בתא, שגורם לכך שאם השינוי שיש בו גדול מדי, התא ייהרס. והוא סיבת המשכב שנרביע בהמות זו עם זו לקיום המינים וכו', והמרכיב שני מינים משנה ומכחיש במעשה בראשית, כאילו יחשוב שלא השלים הקדוש ברוך הוא בעולמו כל הצורך. ויחפוצו לעזור בבריאתו של עולם, להוסיפו בריות. והמינים בבעלי חיים לא יולידו מין בשאינו מינו. הרמב"ן כותב שכאשר מייצרים מין חדש, שהקדוש ברוך הוא לא ברא בעולם, המינים האלה הם סטריליים, הם עקרים, כמו הפרד, כן? שכאשר מרכיבים סוס בחמור, מקבלים מין עקר. אפשר לקחת את הרמב"ן לרמה מודרנית יותר, ולומר שלמרות שלא מייצרים דבר עקר, אפשר לייצר דבר מסוכן. הקדוש ברוך הוא ברא את הבריות בטבע בשיווי משקל, וכאשר אתה יוצר דבר חדש, אתה חושב שאתה משפר נקודתית, אבל אין לך את הראייה המערכתית, הכוללת, כמו שאומר הקדוש ברוך הוא לאיוב, מי אתה, איפה היית ביוסדי ארץ, כך מתחיל הנאום של הקדוש ברוך הוא לאיוב, איפה היית ביוסדי ארץ, מה אתה יודע על העולם, ולכן אל תיגע, דבר שאתה לא יודע, אל תיגע. האם זה נכון? וכשהתחיל הנושא של הנדסה גנטית, הרבה ציטטו את הרמב"ן הזה, ואמרו, גמר, אסור לעשות בניגוד למה שפרופסור גנום שהכל מותר, הם אמרו שהכל אסור בלי שום ספק. כותב הרמב"ן יותר מזה, שגם איסור הזריעה של כלאיים, זאת אומרת שאני זורע זרעים מעורבבים, גם שם הסיבה שיהיה הרכבה גנטית ויצמחו צמחים חדשים. פרופסור פליקס שכתב על הדבר הזה כותב שהתפיסה הזאת שצומח מין חדש כאשר זורעים זרעים יחד, מה שבאמת לא כך, התפיסה הזאת באה מתוך הניסיון של החקלאים היוונים והסורים, אומר דבר מתקבל על הדעת במיני הקישואים והאבטיחים, שכאשר זורעים מינים שונים יחד, יש האבקה בין הפרחים של המלפפון והאבטיח, או בין הפרחים של המלון והקישור, והם נוצרים פירות חדשים עם זרעים חדשים. עכשיו הוא כותב יותר מזה, האיסור לחרוש בשור ובחמור, מפני שדרך כל עובד אדמתו להביא צמדו ברפת אחת ויבוא לידי הרכבה.